I introduce you, maybe we will have more people joining. So welcome everyone to Theoretical and Computational Biophysics seminar. It's my pleasure to welcome our seminar speaker, Professor Phil Biggin from Oxford University Department of Biochemistry. Before I introduce Phil, I would like to just remind everyone to please turn off your microphone. And if you feel comfortable, turn on your camera so it gives a more interactive feel to the seminar. If you have questions, please save them for the end. At the end, I'm going to ask you to raise your hand or type in your question in Q&A, and then actually I'm going to call you to ask your question yourself. So save it to the end. If it's really, really burning question, I hope that our speaker doesn't mind to be interrupted once.
Yeah.
So with that, so let me introduce Phil. Phil received his initial training, I believe, in Oxford in computer-aided chemistry and a doctorate in ion channel biophysics. Then he moves across the Atlantic and does a postdoc at Salk Institute in California with an award that he received actually from Wellcome Trust Prize International Traveling Fellowship. And then he returns to Oxford in 2000 and joins the faculty and goes through the rank. And since 2016, he's a full professor in the Department of Biochemistry, University of Oxford. He is a chartered chemist. He is a member of biophysical societies, both in England and in the US. And he's also a fellow of the Royal Society of Chemistry and a founder member of the Computational Chemistry Kitchen, or CompChem Kitchen, that actually organizes events that are open to everybody, academia, industry, all sorts of aspects of computational chemistry. And he is also currently the chair of Molecular Graphics and Modeling Society. So he research-wise, actually, he is interested mostly in receptors, receptor dynamics, membrane proteins, and how they are interacting with ligands and how they are affected by interaction with ligands. Very important in terms of drug discovery. But today we're going to hear about cellular trafficking. I see in your title, Phil.
Yeah.
So thank you very much again for accepting our invitation and giving us a seminar. The floor is yours.
Okay, thanks. I'll share my screen first.
Perfect.
Can you see this?
Yes.
Okay.
Yeah.
Okay,
Please.
great. Okay, great. So, yeah, thank you very much, Imad, for the invitation, and thanks, Donna, as well, for organizing it in the background as well. It's a great pleasure to talk to all of you guys. I have visited Urbana-Champaign before, many years ago, actually, in, I think it's 96. And there was a lot of snow on the ground. About 2 or 3 feet, I think, actually. I drove down from Chicago and just about made it before a massive snowfall. But I assume there's not any snow there now, is there? But I don't know.
Well, I mean, it's pretty nice. Actually, it gets very cold during the winter. We should get you back here, hopefully soon.
Well, bizarrely, there was 2 centimeters of snow on the ground this morning here. So I think I had enough snow. But anyway, yeah, as Imad said in the introduction, my lab is very interested in using computational methods to look at various aspects of biology, and typically that's involved looking at receptor proteins and channels. But more recently, we've sort of divulged a bit into some aspects of cellular trafficking, which is a very new area for us, and but I think it's quite exciting. And I thought I'd talk about this because it's quite a nice example, if you like, of going right down from using a little bit of QM right up to sort of how that relates, or we're trying to relate that to what happens in whole cells. And I'll show you some data from all aspects of that kind of gamut of techniques. So the system I'm going to talk about is something called the KDEL system. And this is one of the major kind of transport processes in the cell, systems in the cell. So it's called the KDEL system because it relies on a tag, if you like, at the C-terminus of various proteins, which I'll come on to, which has a lysine, an aspartic acid, a glutamic acid, and then a leucine residue, so hence KDEL, right? And the basic idea is that in cells, they have this kind of problem that they have to get various proteins synthesized and made and processed, and then shipped out basically to various other locations in the cell, maybe the plasma membrane, or secreted, or indeed other bits in the cell, like various vesicles and things. And in this diagram, this kind of summarizes what kind of goes on. So proteins start off typically in the endoplasmic reticulum, which is on the left here. And then they'll go through a kind of maturation process where they'll make their way towards the right, towards the Golgi. And in this schematic, these kind of proteins that are going to move are kind of shown by these yellow asterisks, yeah, these yellow stars, sorry. And that sounds great. And what happens is that they get, basically, they start off in the ER and they get picked up by a kind of COP2 complex forms around or starts to form a vesicle here. And that vesicle then buds forward and then moves it towards the tubular cluster and then eventually on towards the Golgi. And that's fine, but a problem with that is that sometimes you can also end up with some of these vesicles containing proteins that we know have to reside in the ER, so-called ER resident proteins. And so they end up in places where they shouldn't. So they end up in the tubular clusters or in the Golgi. And the cell has to somehow deal with that, has to get them back into the ER. And the way it does that is that it relies on these proteins having this tag, this so-called KDEL tag, which is recognized by a receptor, the KDEL receptor, which is this green guy, okay? And when that binds or recognizes a protein like is shown here, where my pointer is showing, hopefully you can see that, this then triggers COP1 proteins to kind of cluster around and form a vesicle, which then allows this retrograde transport of the ER resident protein in complex with the KDEL receptor back to the ER, where it can then release the ER resident protein and the whole process can start again. So it's a kind of rescue system, if you like, for bringing back proteins that have been mistransported, if you like, too far out to the Golgi or the tubular clusters. And most of this has been mapped out for a few decades, actually, mostly by people like Hugh Pelham in the 90s. But actually the details of how this is controlled or how this takes place is still remarkably unclear, actually. One thing you'll notice that I've indicated at the bottom here is that one thing we do know is that in the ER, the pH is about 7-ish, whereas when you get to the Golgi, the pH now is a bit lower, it's about 6, something like that, okay? So people have speculated that that may be some part of the process in terms of controlling how these KDEL receptors actually function. So that's the basic cell biology background, if you like. Hopefully that's clear enough. And the way we became interested in this was from a great piece of work from my neighbor, literally in the lab. He's next door but one in office space down the way. And his name is Simon Newstead, and he's a crystallographer. And he solved the structure of this KDEL receptor, both in the apo form and in complex with the KDEL peptide. So this is just a peptide of that, which you can see being docked into just a simple morph that was between the two states. And there are a couple of rather interesting features from this structure, which required a little bit of further investigation, at least we thought they did anyway. One rather interesting feature that was first noticed was that at the bottom of the binding side of the peptide, where the peptide binds, which is going to be zoomed in on here on the left, you'll actually see that there's a short hydrogen bond being formed by this tyrosine and this glutamic acid, okay? And you'll also notice the presence of histidine here, this histidine H12, which you're probably thinking, well, that could be a proton sensor. That might be involved some way, possibly in controlling the way that the receptor works. So that kind of piqued our interest. And from that, there are lots of interesting questions which are kind of perfect for a kind of computational approach to look at these kind of problems. And this is all work that I'm going to talk about has been done by William in the lab, who was a graduate student at the time. He's now since graduated and is a postdoc in the lab. But William did all of the work, and he deserves all of the credit, and I deserve all the flames or criticism. So there's a number of questions that one can ask, and they start from simple questions like, is this short hydrogen bond really there? Sometimes these short hydrogen bonds are sometimes called low barrier hydrogen bonds, meaning that it's very easy to move the proton from one acceptor oxygen to another. And we can ask certain questions like, what are the energetic consequences of having such a short hydrogen bond? What does that mean? Does it mean anything at all? Could that H12 be a pH sensor? And how would that affect the short hydrogen bond, if at all? Okay. Does the H12 histidine affect the behavior of water in the surrounding pockets? And I'll show you a bit more detail of that as we go along. How does the peptide engage or disengage from the binding site? And how is selectivity for that actually controlled? And can you explain that from the structure? And I'm sort of preempting to talk a little bit here by lining you up to expect that it's not going to be readily explained actually by the structure. There's an interesting little twist at the end, hopefully. So in terms of the talk, this is a basic summary of the outline of that. I'm going to sort of go from examples of where we've tried to use quantum mechanics right through to whole cell measurements to try and address some of these questions. And I'll divide the talk into kind of three sections, okay? So first of all, I'll focus in on the sort of very fine minutiae details of the first KDEL structure and what we can learn from that. And then I'll kind of pivot a bit to think a little bit more about the role of pH and what that might mean in terms of the function and the structure of this receptor and what it means for the biology of it. And then at the end, I'll kind of change gear again, and I'll talk a little bit about how we think the signal is recognized and what the implications are for that has for the in vivo processes. And as you'll kind of gather as we go along, it's by no means complete. There are many questions that you'll see that are unresolved, even though I'm going to tell you quite a lot of things that we think we have resolved. There are lots of things that we definitely haven't resolved as well. And hopefully you'll see that as we go along. So let's go back to this short hydrogen bond, just in a little bit more detail. So here we're looking at the KDEL bound structure on the left and comparing that with the APO and just zooming in on this hydrogen bond between two residues at the bottom of the binding pocket. And in the KDEL bound state, this is actually rather unusually short, the distance between these two oxygens compared with a more normal distance that you might expect these, if you were to examine any PDB structures where you'd see them in this kind of proximity, you'd expect them to be about 2.8, something like that. So 2.5 is really quite a small distance. So if it's real, are there any kind of energetic effects of that? And does protonation state of H12 influence that in any way? So first of all, though, we have to actually confirm where the protons or proton likes to actually sit, because of course in the crystal structure, we typically cannot see that. So the first thing to do is try and just reassure ourselves that we can see where the protons like to sit. And you can do that in a very simple way. You can just do that with a minimization protocol with using quantum mechanics kind of method. So the idea is just to systematically position the proton on all of the oxygens and then just minimize it and see where it goes, okay? So in the apo state, okay, you put the proton on near the tyrosine. Yeah, sure, it minimizes to the tyrosine. put the proton near the OE2 atom here of this glutamic acid, sure, it stays there. And if you put it on OE1, no surprises, it also just then pings onto the OE1. So that's entirely as expected. The interesting thing comes, though, when you look at the KDEL bound state, because in this case, if you try to put a hydrogen near the OE1 oxygen, it actually won't stay there. It'll actually minimize onto the same position that's near the O of the, or the OH group of the tyrosine. So you can't seem to put a proton on this OE1. It will always minimize down onto the OH of the tyrosine. So that's the kind of first clue that this hydrogen bond, it really is a short hydrogen bond in terms of the geometry at least. So a hint, effectively, that it's a low barrier hydrogen bond. So we can do a little bit more in-depth calculation for this, just to convince ourselves that this is true. So if we go back to the apo state, and what I'm trying to show you we can do here in this calculation is to basically compute the energy for transferring a proton from the tyrosine OH, so this would be here, this location, and then just moving it, just linearly interpolating the position of the proton, calculating the energy at each point, as if you put it onto the glutamic acid or the glutamic acid. And again, what you see is this is the classic profile. You see there's a well, and then there's some sort of barrier, and then you go into a well again. Okay, so this is entirely consistent with those minimization results that I showed you in the previous slide, right? So if you started somewhere here, you just go down the well into the nearest minimum. Okay, so that's entirely normal. We can do that calculation in the presence of different protonation states of the histidine. So you can either put the proton in a neutral histidine on the delta nitrogen, or you can put it on the epsilon nitrogen, or you can, of course, protonate it completely and put it on both the delta and the epsilon nitrogens. What happens if we do the same calculation with the KDEL bound state? Okay, so the same exact calculation, same type of calculation, just moving the proton, calculating the energy at each point. And now you can see something a little bit more interesting, perhaps. So here you can see that actually you have a little well, but then it tends to flatten out. And this flattening out here is really prominent when you have a protonated histidine. So this is the green line here. You can see here you have a little well, and then it's really flat. And just for a little bit of background information, these dashed lines you can see here, this is the so-called 0-point energy. And that is the energy basically associated with an OH vibration. So essentially, this is about the same energy here as an OH vibration. So it's pretty flat. So there's no real barrier. So again, that kind of supports the idea that this is a low barrier hydrogen bond, and moreover supports the idea that the protonation of H12 influences that process and really kind of favors it, right? So that's all nice. And then just the last kind of kitchen sink for this kind of calculation. We then decided that we just check this with using QMMM calculations, and this time trying to do a kind of free energy calculation whereby in the apo state, we either try to stretch the bonds to shorter different distances. So you remember, hopefully in the apo state, that the distance between the two oxygens is larger, sorry, than the KDEL bound state. So if we try to squeeze them together, so we'd start here and then we'd move left. If we try to squeeze them together and compute the free energy, you can see that it doesn't really matter whether you protonate the histidine or not. The energy rises very, very sharply. Okay, it doesn't really want to do that. In the KDEL bound state, you get so it's the opposite kind of calculation. We start off in the bound state, and this time we're trying to stretch that bond. And we start off here, and then we try to stretch the bond and plot the free energy as a function of that stretch. And you can see here, there's a distinct effect of the protonation of the histidine in this case. And indeed, when you have it protonated, you have this green line, which really makes it want to sort of stay essentially in this small distance. Okay, so the protonation again of histidine is really favoring you sitting in this well. Okay, that maybe sounds maybe looks a little bit counterintuitive, but it does actually make consistent sense with all the rest of the data that I've shown you. Okay, so hopefully I've convinced you that the QM, from the QM point of view at least, we can argue that this really is a low barrier hydrogen bond. And the key thing about low barrier hydrogen bonds is that they usually are a lot stronger than the normal hydrogen bonds. So that might be relevant for stabilization of the KDEL peptide being bound. I'll come back to that towards the end. Okay, yeah, so the APO cannot form a short hydrogen bond, and the KDEL bound, well, there's more energy to do that separation. So you can also ask the inverse question, right? So far we've asked, how does the protonation state affect the short hydrogen bond? But we can also spin it the other way around and say, does the short hydrogen bond, or the presence of that, affect how easy it is to protonate the histidine? Okay, and this is a little bit of a complicated calculation. There's a delta G of deprotonation of H12 at the same time that we protonate H12 in the apo state. This is probably a lot to swallow in one go. I don't expect anybody to particularly gather it all necessarily, but don't worry about this. I'll explain what the take-home message is in just a second. We do this in quite an efficient way. We actually do 2 free energy calculations in the same simulation box, and this gets around a lot of problems. So essentially, we're deprotonating H12 when the KDEL is bound. So you might be able to just about see the pink KDEL bound here in this system. So we're going to deprotonate H12 in this system, but at the same time, we're going to protonate H12 in the apo state, which is up here. And the reason we do this is it's quite efficient. That's one thing, but it also gets around problems of charge annihilation in free energy calculations, because we actually maintain the same amount of charge overall at all times. Anyway, the key result of this is that basically these results here, where we have a short hydrogen bond imposed, it basically says that it's energetically unfavorable to deprotonate H12 in the KDEL bound state. Okay, it requires this amount of energy. So we'll repeat this 3 times, you get fairly consistent results. Okay, we have to impose a restraint, because otherwise the system will want to move towards, a non-short hydrogen bond, because of course most molecular mechanics, which is what this calculation is, are optimized to not have short hydrogen bonds. They're kind of parameterized effectively to give you, fairly normal looking hydrogen bonds. So you start to see that actually, these numbers here will look a bit less than if you keep the hydrogen bond restricted. I wouldn't worry about too much about that. That's just a little technical point. We can also do a couple of control restraints just on random histidines, just trying to change the protonation of those and see what the effects of those. And you can see here, there's maybe half or up to maybe 1 kcal mole difference, but it's essentially a lot less than this amount of energy that you see here. So the take-home message there is that energetically, it's unfavorable to deprotonate the histidine when the KDEL is bound. So where does that energy come from, right? So that sounds maybe interesting, but where does the energy come from? And if you remember from that little movie I'm showing you on the left and just repeating it, there are some key changes that happen when you move from the apo state to the KDEL bound state. And you can see probably this helix is one of those changes. But another key change that's maybe not obvious from the movie is that there are water pockets that exist in the apo state and in the KDEL bound state. And these are shown by this gray surface here and this orange surface here. When you bind the KDEL peptide, you actually reduce the volume of water in the gray pocket, if you like. You can see that over here. And also in the orange pocket, and you also get the formation of a small little red pocket here as well. So I'm going to explain what that means. The first thing is that the KDEL binding reduces the cavity on either side of the hydrogen bonds. That's the first key observation. But it also means that there's a slight difference in the way that the water behaves on either side of these as well. So if you then do grand canonical Monte Carlo, which is a way of computing the energetics of putting waters into pockets, that's typically what we do. And when you do that under different protonation state conditions for this histidine, in the apo state, it doesn't make any difference. Okay, the water networks that you see in these pockets are very, very similar, almost indistinguishable from each other. But when you put it into the KDEL bound state and you do the same kind of calculations, then actually there's a very distinct difference when you have the histidine protonated compared to when it's in its neutral state. And when it's protonated, you actually favor a lot more water chaining kind of happening in this bottom pocket compared to when it's in the neutral state. Hopefully you can see that in these two different purple circles, which are highlighting the main difference. So when it's protonated, you get a nice chain of water molecules forming, which is obviously aided by the protonation state of age 12. And we think that's actually going to be quite important for understanding the link between all of this in terms of the hydrogen bond formation and the KDEL bound state. One other thing to note is actually you can see there are two water molecules that end up being bound here as well, next to R159, which makes a key interaction actually with the KDEL bound peptide as well. So we think all of this is very important for stabilizing the KDEL bound peptide. So, okay, so I've told you a few things, and let's just pause at this point and think about what does that actually mean so far for how we think the KDEL receptor works in the cell, okay? So the idea is that we think you start off in the Golgi, remember on the right-hand side of that first slide I showed you. And if you remember, there's a slightly lower pH in the Golgi, okay? So we think this histidine will likely be protonated, okay? Then you have KDEL binding, okay? And then this forms a short hydrogen bond. We don't quite know whether this is a chicken and egg kind of situation at this stage. This short hydrogen bond forms somewhere. It may be that you have to form that before you bind KDELs, or KDEL binds and then that forms the short hydrogen bond and it helps to clamp down on the KDEL. And then you get this kind of separation of water pockets that happens at this point. Then there's a process now which means that this will then will pick up COP2 and will then be formed into a vesicle and will then be transported back into the ER, okay? And when we're in the ER, we're back up at higher pH. And so this histidine will want to become deprotonated, okay? That will then destabilize the short hydrogen bond, okay? And that will then allow the KDEL peptide to be released back into the ER. And then you can begin the whole process again. This can then be shuffled out to the Golgi, and then you can pick up more KDEL receptors. So that's how we think the short hydrogen bond and this histidine sort of collude, if you like, to use pH to ensure that you pick up KDEL in the right place. Okay, so that's one aspect. Okay, I'm going to switch gears a little bit, and now I'm to talk a little bit about how we think the signal is actually recognized. This is kind of a little bit interesting. So what we did first was just to do a kind of series of steered MD. I'm sure many of you in the audience are kind of familiar with this process. And we can do this, we actually did this backwards, so an unbinding event, but I'm just showing you in reverse, just so you get an idea of how it might actually bind. And you can kind of see that there's a lot of free carboxy acid, amino and carboxy groups that kind of talk to this ladder of arginines that's in the binding pocket. And that's a kind of like a Baton-Relay system, if you like. So, basically the first carboxy group talks to the first arginine, and then that kind of hands it on down to the next arginine, and so on and so forth until it gets fully bound. And you can kind of see that quantifiably here in this graph. So the purple line is the free energy of that process. So here we're fully bound. So here would be outside the binding pocket. And then you can see we gain about 20 kcal of free energy. And as we do this, you can see there's kind of a nice handing off of R169. So that makes the most interactions when we're in this position. So that's this guy over here on the right. And then it kind of hands over to the next one down, which is R5. And then finally it hands over to R47 before then engaging with R159 as well, which I'm not showing you on this graph. Keep it reasons of clarity. So we kind of understand what's going on in terms of this kind of pattern relay. The next question though is, how does this relate to the signals that we see in the cell? And although so far I told you that most of these peptides or these proteins that are going to reside in the ER have a K and then a D and then an E and then an L. It turns out that actually, if you look at all the proteins that have to be retrieved in the cell, then the frequency of, there's quite an interesting pattern comparing the frequency with abundance in the cell of these signals. So this is one of these kind of conserved residue plots, if you like, of the last 7 residues in this case. So here is the DEL, you can see that. And then you can see at this -4 position, we have, well, you know, it can be a lysine, or it can be a histidine, or it can be an arginine, and very occasionally a glutamine as well. So, and K and H and R have approximately equal frequency, it looks like. But actually the abundance, meaning the total concentration of proteins in the cell, if you look at that, then it's lysine that predominates, okay? And you can see that more clearly perhaps in this plot in part B of this figure, where you can see here we're just looking at HeLa cells and some mouse brain cells. And you can again see that KDEL is by far the most predominant protein in terms of its abundance in an actual cell. Now, there's a curious thing here, because actually, if you then look at the affinity of the KDEL versus the RDEL versus the HDEL, then there's a rather interesting pattern here. So KDEL this is like a competition experiment, so just competing off things, and you can see here KDEL is basically black, and it has very similar kind of profile of affinity to RDEL, right? So not much real difference there. DDEL, okay, we can make that and we can look at how well that binds, and obviously that does not bind very well at all. That's not maybe surprising, considering it doesn't really turn up in this pattern. And you'll see later why that's obviously not going to be a good binder as well. But the curious thing is that the histidine, or HDEL, has by far the best binding affinity, okay? And that's a little bit curious. We didn't really understand why that would necessarily be the case. So the take home from this slide is that affinity is not necessarily correlated with the abundance in the cell. So what does that mean? So we could ask, the first question we said, well, it could be that maybe the receptor has a slightly different conformational change, right? Maybe it has a slightly different conformation when it binds histidine or HDEL compared to RDEL or KDEL, okay? And it turns out that's not the answer. So we did, Simon did a lot of structures of this. So this is looking at HDEL, this is RDEL, KDEL, and you can quite see with your own eyes, just by looking at these three, that they're very, very similar in terms of their conformation. And just to convince you further, if you overlay them all, they're pretty much right on top of each other, okay? So here are the different positions. This is the L at -1 and so on. And you can see here, this H or R or K basically pretty much sit in the same place, okay? So it doesn't look like there's any difference in the conformation of the protein that could account for this difference. And if you look carefully at some of these, you can see some rather interesting features of the binding site, which I'm going to expand on. So if you look at, for example, H or KDEL bound, you can see that there's a couple of residues that kind of hopefully stick out to you as being particularly important, maybe important. One is this E117, okay? This looks like it should be super important for binding because, it's talking to this histidine and it's talking to this lysine, so it looks like a classic sort of bridge there. And if you remember, I told you that the DDEL wasn't a very good binding, and now you can begin to rationalize why that might be the case, because if you put a D here as well, then that's a negative charge talking to a negative charge, so it's not going to be very happy, right? And the other thing you might notice is that there's a nice interaction, kind of planar interaction between the histidine here and the tryptophan 120. From this alone, it's not that obvious why histidine would be a better binder than lysine or arginine in that position. So we think it might be to do with pi-pi stacking. That might be one explanation. But there's another question as well at this point, which is this histidine here in the signal likely to be protonated as well? Again, we can ask or use computation to kind of address these kinds of questions. It's a classic sort of problem for computation to provide some insight to. So that's the first thing to do, is ask the question, is the histidine of HDEL most likely to be protonated? And you can just show this, well, you can convince yourself just by looking at fairly simple, short molecular dynamics simulations of either neutral histidine, protonated histidine, and then comparing it to a lysine. Okay. And what you can see, hopefully, from this is just some arbitrary distance that just measures the distance of the histidine or the lysine nitrogens to probably I can't remember if it's the center of mass of the oxygens of the E11 set. And what you can see here is that the histidine, when it's protonated, is pretty rock solid, right? This is 3 repeats you can see here. Not very long simulations, but it's pretty convincing that this looks pretty similar to the case with lysine there. And so if you have deprotonated histidine on the epsilon nitrogen, you can see that it's wobbling around quite a bit as well. Now, one slightly unusual thing is that if you put the proton on the delta nitrogen of the histidine, of the signal, then it's actually, a lot further away. So it's like 5.5 compared to say 2, but it seems like it's quite steady. It's quite sort of, you convince yourself that's pretty rock solid there, actually. Yeah. And the reason is that actually what happens is that a sodium ion comes and sits in there and helps to stabilize the interaction between the histidine and the glutamic acid. So that's a little bit of a surprise. We don't know whether that's real or not at this stage. Difficult to really test experimentally, I would say, as well. Now, the hist being protonated, we then got confirmation that it was likely to be true, because you can confirm that by doing those binding experiments at different pH. And again, looking at the curves, you can convince yourself quite nicely that low pH gives you much stronger binding compared to higher pHs. So pretty sure that histidine is going to be protonated. Okay, so then we started to worry a little bit about the histidine interaction with this tryptophan underneath, tryptophan 120. So again, try to do a bunch of free energy calculations just using molecular mechanics, and just trying to look at different kind of transformations, essentially, just to try and quantify this. So the key one is actually this bottom one, where we're basically computing the free energy difference to change from a KDEL to an HDEL bound peptide. Okay, and that's actually, we can actually do that in quite a complex way, which I'm not going to bore you with, but we can take into account all of the his protonation states by sort of weighting them correctly and so on. So, and you can see here that actually even molecular mechanics is able to pick up that the HDEL is going to bind by about 2 kcal per mole better than the KDEL bound state. Now, so that's quite nice to see the molecular mechanics and pick that up. And if you mutate W120 to an F or an alanine, then you actually see that actually this starts to drop away, and you don't get actually any preferential binding. So yeah, so these are the key results. You can see that we're able to pick up this kind of sensitivity to H rather than K in molecular mechanics. And we can then actually also just check with quantum mechanics, because we can't delve too much in terms of pi interactions and so on and so forth, but we can do that with quantum mechanics. And what we found is that actually when you have a protonated histidine, you obviously get nice cation pi interactions. That's basically what this induction effect here is showing you. You get quite a nice contribution from that. But you'll notice also that the dispersion effects are also quite large in terms of the energetic contributions there. So you can convince yourself that, yeah, actually this cation pi, or these pi interactions between histidine and that W120 are actually rather important, and probably are the reason that you get this gain in affinity compared to, say, for example, KDEL. Okay, good. So now, if we then go back and compare that to a series of mutagenesis, then these 120 calculations match rather nicely the mutagenesis data. So just looking at the cyan, So this on the left is looking at HDEL peptides binding, and this is looking at KDEL peptides binding. And you can see here that, yeah, actually, when you look at the cyan bars, there's lots of other mutations here that I'm not telling you about, but if you look at the cyan ones, that's one I want to focus on. You can see here, yeah, there's a nice reduction in binding for HDEL, and it's actually even worse for KDEL as well. So you really do that tryptophan really is very important for the binding of these peptides. More so, it turns out, than the E117, the glutamic acid. If you remember that at the E117 position, which is at the top, which we thought was going to make a nice salt bridge, right? And so this is kind of confusing, or on the face of it, looks a bit odd, because actually, if you mutate E117 to an alanine, for example, so getting rid of the charge of the salt bridge, pretty much completely, then actually in the HDEL system, or HDEL pep binding assay, you only really knock it out to about 80% of wild type. Yeah? And that's true actually in KDEL as well, maybe a little bit lower. But you can see here, these mutations for this glutamic acid are rather puzzling, because you would anticipate that they would actually give you a more dramatic effect, considering their likely interaction on the basis of the structure. So it's not obvious from this, actually, and the binding pose, why that E117 is so important, and it is very important. It's conserved amongst KDEL. So I'm going to come back to that later and explain how we think this works, actually. So just back to the affinity versus abundance question. So remember, I showed you this before. This is the frequency, and this is the abundance in the cell, and this is the affinity, and reminding you that histidine has the highest affinity. So it seems a little bit strange that the KDEL receptor itself is apparently optimized to bind HDEL tags, right? Because it has the highest affinity for HDEL tags. But the abundance in the cell clearly tells you that most of the proteins actually have a KDEL tag. So that seems a slightly strange paradox in the biology here we should try and address. And you could ask yourself, well, does it actually matter in the cells itself? Do they actually care, right? So this is an experiment done by, a lot of experiments, I should say, done by Andreas, who's in Francis Barr's laboratory, also in the department. So this is a giant collaboration between three big groups in biochemistry. Anyway, what these guys did was that they generated N-scarlet fusions with these different signals, and then looked at the steady state distribution of KDEL receptors in cells, okay? So what that means is if you don't have any ligand, then the KDEL receptor, you can see that it likes to hang out in the Golgi, right? Because it's not doing any retrieving, essentially. So that's kind of your kind of control point. And then if you add back proteins that have these tags at the end of them, so KDEL, RDEL, or HDEL, then you see that actually these get pulled back to the ER under steady state conditions. This is a steady state comparison to the ligand, no ligand setup. And you can see that these are basically in real life cells, the behavior is pretty similar between KDEL, RDEL, and HDEL. So it doesn't really seem to matter. You can also do the same experiments with ADEL peptides or DDEL. And you can see here, these are not very well driven back to the ER at all. Actually, they're actually ends up being kept in the Golgi, which is exactly as you'd expect, because those aren't expected to bind very well to the KDEL. Okay, so in the cells, doesn't really make any difference, apparently. So that's a bit curious, right? So you could ask the question in a slightly different way and pretend, well, is there a problem with high affinity? Maybe there's an issue that if everything had high affinity, perhaps there's an issue. In other words, why are the signals not all HDEL? Let's think about that problem. And actually, that's a question that you can address in terms of biology, because you can do an experiment where you can say, let's pretend that the signals are all HDEL, right? So this is a bit of a complicated slide. I'm not going to really go into all of the detail. In essence, what we're trying to do here is pretend that we're competing with HDEL, okay? And what happens is if you have a lot of HDEL peptides, okay, once you have a lot of HDEL peptide present, and then you ask how well does your resident ER protein, so BIF, which is classic ER resident protein. It has this KDEL signal. These are some other ER resident proteins that have different signals. And then you basically are saying, how well does this compete with these different signals? That's the question we're asking. And what you can see here is that if you're the one that sticks out is this one, right? So if you say, you look at the HDEL, so you add a lot of HDEL competitor peptides, and you can see here that now a lot of the resident ER proteins actually end up being secreted, okay? So instead of these proteins being recruited back to the ER, if you have too many HDEL peptides hanging around in the Golgi, these basically cannot compete with those HDEL. These guys cannot compete with the HDEL peptide. And so they end up being secreted instead of being pulled back into the ER. So if you have too many HDEL binding peptides, you basically end up poisoning the system, okay? So that's kind of the rationale we think behind it. So the essence of this is that essentially the retrieval system is selective, but it's not optimized for binding affinity. So this is a rather strange concept in many ways, okay? It's actually optimized to retrieve a broad range of different proteins of differing abundance, okay? And you need the HDEL. This is the clever bit, if you want, because you need the HDEL, which has a higher affinity. You need that for the lower abundance proteins because there's less of them hanging around. So you need some way of making sure you can fish those out when you need to. And so as long as they've got the higher affinity signal, then you will successfully retrieve them, right? So that's what we think is actually happening. Okay, let's zoom out a little bit more, okay, and consider the broader problem. So the KDEL receptor, if you think about it, has to fish out these sequences from a massive pool of proteins floating around in the Golgi, okay? More specifically, it has to find KDEL or RDEL or HDEL from everything else that might be floating around, okay? So that's the challenge for it, okay? And how does the receptor actually do that? Particularly, as I just told you, that E117 doesn't seem to be particularly as important for binding as first suggested, yeah? And this is conserved residues. So what is its role actually in this whole process, given that doesn't seem to really change the affinity of binding the peptide? Right? So what you can do is actually what all biologists tend to do, right? Is go and look at sequences for clues, okay? So that's what we did. So you look at the KDEL receptor sequences, okay? And here's all the classic ones. So there are actually 3 different KDEL receptors in humans. But they're all pretty similar. And so we're looking at a few classic ones here. So mouse, chicken, etc., fly, and so on and so forth. And you look at key positions, which is what we're doing here. And if you notice, the key ones are at the bottom here. Okay, so everything else up here is more or less the same. Just focus your eyes on these 2 bottom ones here, which are yeasts, you'll notice. And also, in yeast, it turns out that they actually do use ADEL and DDEL as retrieval signals. Okay, so that's a slight, that's an interesting quirk, if you like, right? And if we look at where the main differences are, then we can see a few key positions. So remember this E117, okay? You can see here nicely always glutamic acid, okay? But in the yeast system, it's not glutamic acid. It's either an N or a Q, okay? And there are another couple of positions here you can see are very different from their positions in equivalent positions in mammals, especially. So what does that mean? Okay, so if you look top down now at the receptor, so this is what I'm showing you here. This is the signal, KDEL signal. Okay, looking top down, here's our E117 friend, okay? Here's where D50 sits, so this is this guy here, okay? And 54 is pretty much next door, okay? So these 2 points here are just here, okay? And then if you show, you can plot a surface and then look at the electrostatic potential that's sort of surrounding the peptide, which is what is shown here. And you can see here, there's quite a distinct sort of shape to that potential in the sense that the bottom half is very positive and the top half is pretty negative. Okay, Now, one thing you can show is that, and I'm not showing the raw data here, but the experiments done by Andreas and Francis show that actually just mutating this E117 to Q or N is not enough on its own to bring back KDEL. Okay, it still doesn't bind, even though you change that residue to asparagine or glutamate. Okay. And the other thing to note here is, which I'm trying to draw your attention to, is that position 50 and 54 are kind of on the periphery here. And they're not really actually, they may look a bit close to maybe this E-5 position, but that's just the view I'm showing you. They're actually at least can be no closer than 5 angstroms between the closest atoms here. So they don't really play a role in directly talking to the KDEL bound peptide when it's actually fully bound. So what does this mean? So we think actually the way to explain this is actually in terms of an electrostatic steering kind of role. So if you look at the original kind of map of the electrostatic surface, it looks like this. And then what you can do is just imagine changing the surface by changing the residues at these different positions to what they are in yeast. So this would normally be aspartic acid at position 50. We change that to its equivalent in yeast, which is an asparagine. Then you can see here we're starting to knock out a little bit of the positive charge here. Okay, electrostatic potential. And again, if you knock out 54, then you can see again we've modified the potential even further. And then finally, if we also do Q or E117 to Q, now you can see actually we've got rid of most of the negative electrostatic potential on the surface protein, and it's pretty much always now positive. Okay? And you actually need all of these 3 residues to change in order to bring or to recruit KDEL peptides. I'm not showing you the raw data for that as well, but just trust me, the experiments back this kind of idea up quite nicely. So that's what we think is happening. We think that actually these 3 residues here actually act in tandem and act like a kind of electrostatic steering device rather than governing the binding affinity directly per se from just what you saw from the binding pocket itself. So the role of this -4 position, so where it's a histidine or histidine or lysine or indeed an alanine or an aspartic acid, is that it's actually a kind of electrostatic steering guide. So just summarize what I've hopefully told you. I told you about this short hydrogen bond. I told you that impacts on H12 protonation, and that actually also works in vice versa as well. Okay, that H12 protonation also affects or influences the short hydrogen bond. I've also hopefully told you that this also controls the behavior of the pockets in the HDEL receptor, and that helps to clamp down on the KDEL signal. I'm sorry if I've gone a bit dark, the lights automatically dim after a time. Hopefully you can still see me. And then an arginine ladder is important in terms of how the peptide actually engages or disengages in that initial process. And then there's an unexpected role of some of these residues in the binding control. In particular, E117 and W120 have unusual roles, which were not obvious from that first structure, but with a little bit of computation and sort of computational insight, we've hopefully shown you what we think is now happening and how to interpret that. So just to summarize what this means in terms of biology, the important thing here is that rather than selecting for the sequence, E117 may actually be more important to select against unwanted signals. Okay? So the way that we kind of imagine this is kind of shown in this kind of cartoon where you have this kind of signal, and then this position at -4 is a little bit like a kind of passkey residue or something like that. Okay, and that's the key interaction of that dictates or it's influenced by these so-called gatekeeper residues at D50 and E117 acting in tandem particularly. And if they're not right, then, or if they're not the right combination, then you won't allow the binding of the right signal for the right receptor, as it were. So we think this is what happens. We think that if it's H or R or K, and it's fine, it gets past the gatekeeper, and then it goes in through through the arginine ladder, and then it gets locked in position, and then it can be then trafficked back to the ER and then released and so on and so forth. And if it's not the right residue, then it's prevented by electrostatic steering. So it's complex, and there's a lot of things that I haven't told you about, and a lot of things that are ongoing, but hopefully this has given you a kind of flavor of a new direction that we're kind of exploring. And hopefully it's given you a kind of a little bit of insight into how we can relate sort of computation back up to all the way up to cell biology, which I think is quite a nice link to make these things. So I'll just end by quickly thanking people and be conscious of time. So Will, as I said, the person who did nearly all of the work, and he deserves all of the credit for this. I must also thank people in Simon's lab and also Andreas and Francis for all the cell biology work. They deserve all the credit there. And I'll stop talking, and I'm happy to ask or answer any questions that you may have.
Well, thank you very much, Phil. Very interesting work. I have actually, let me start with my own question. So it goes back to your last slide about this sequence and the signal sequence. So this DEL signal is kind of unique for the proteins that are tagged to interact with this with your receptor. Is that?
Yeah, true? Yeah. The signal is unique depending on which. So depending on whether it's an ER, whether it's a protein that has to live in the ER, and also which species it's from. I see. KDEL or ADEL in the yeast. Yeah. But in mammals, it's KDEL, HDEL, or RDEL.
Yeah, it's amazing to me that no other kind of segment has, or it should be at the end of the protein, obviously, doesn't have this DEL. So, but
yeah. It's kind of curious that if you look carefully, those sequences are very specific for the resident the ER resident proteins. Yeah.
And I have actually another kind of, I really found the double bilayer that you use for your free energy calculations very cute, actually, to change one to protonation, the other one to deprotonated form. So do they have to be in two bilayers, or we can put them in the same bilayer, I guess? Should be possible?
Yeah, that's a good idea. Yeah. No,
because I find it very useful, actually, trick to have two copies. Of course, your system is going to be larger a little bit, and you have to simulate more. But it's a very neat way of taking care of that charge difference and change.
Yeah.
And not worry about.
There's no reason you could. You're right. So there's no reason you couldn't do it in the same bilayer. Actually, yeah. Okay. We just did that because of the. I guess we didn't think about it because we just took inspiration from the conductance.
I see. Yes. Yeah.
I mean, thought that we could just use that, and that exactly. So we didn't actually think. You're right. We didn't think very carefully about that.
Okay, maybe actually the dimensions of the box would be more smaller if you.
Yeah, they might be. Yeah. I don't know. That's a good point.
Cool. Shashank, you had a question?
Yeah. I think it was partially answered, but still, I think I can ask.
Sure.
So thank you for the talk. It was really interesting. So I have a specific question in the first summary of the first part, where you mentioned, or essentially you were giving us a picture of the protonation of histidine and how it is coupled to the short hydrogen bond. So in that schematic representation, you also showed that the proton is already coupled to histidine. And so essentially my question is, do we know the coupling between or when this short bond forms, and or the sequence of events, to be more precise, of events on the protonation and the formation of short bond?
Yeah, so the answer, yes. So yeah, you spotted the hole. So the answer to that is no, we don't know we don't know the exact dependence or the sequence of events. So it's a kind of, I guess the assumed sequence of events is that the histidine H12 is protonated, and that then favors the binding, or more readily enables the binding of the KDEL peptide. But we don't know that for sure. You're right, it could be the KDEL binding helps to then ensure that the histidine gets protonated. But if you think about the biology of it, I think that's the way I would kind of rationalize it, and thinking, well, when we're in the Golgi, we're lower pH. And so probably that histidine, we haven't done those calculations. You could probably try and calculate this, but the histidine you'd expect that to be more likely to be protonated. And that would happen much faster than presumably any KDEL binding event. So that would presumably happen first, and then the KDEL binding event would happen. But we don't know for sure. That's a good question. Yeah.
Okay. So I also had a kind of related question to that region. So I noticed you talked about wetting and de-wetting of that region during the process. So did you have water molecules in that region when you did your QMM calculations, Phil?
Not so for the QMM, yes, there were water molecules present. Yeah.
But not for the initial QM kind of.
Not for the initial calculations. Yeah.
Fair enough. Okay. Cool. Any other questions? Again, you can raise your hand, or actually we are a small group now. You can just start talking. Any other questions? If not, let me thank one more time. Phil, thank you very much for being with us so late at night and your time and giving us a seminar. So you're using, and I wish everyone a great week and great time. Okay, so everybody can leave now. So you're using Keynote, right, Phil?
Yes, Keynote, yes.
So how did you use that red cursor or pointer?
Oh, I have an iPad.
Ah, I see.
You're using an iPad as the remote, then you
very
can
nice, very nice.
Cool. Yeah, it's a bit annoying that you can't just use a
yeah, in PowerPoint, people have that kind of pointer.
Yeah,
but in Keynote, I was wondering how you did it. Okay, so you're using my iPad. I should practice with my son a little bit to learn that. Thanks again, Phil. Very nice of you.
Good to see you. Ciao.
Thank you, guys. Thank you. Nice to meet you. Nice to meet you earlier as well.
Hi. Bye-bye.
All right. Thanks.
See you.